Welcome back to my channel Bio World once again. Today again with a very interesting topic of photosynthesis, that is C4 cycle, carbon 4 cycle of photosynthesis, which is also known as Hatch and Slack pathway. Now this is a very important cycle in terms of the plants which remains in dry, hot and high light intensity climate. So plants like maize, sorghum, amaranth, these are some plants which show C4 cycle. But remember everyone that C3 or Calvin cycle is the most common one, and it is the most important one as it runs in all of the cycle, whether it is C4, C2 or it is C3, any cycle, camp cycle. C3 is the main cycle which has to go through the every cycle to produce glucose. Now in this cycle, C4 cycle, the plants which shows C4 has a little characterized anatomy of their cross section of the leaves. You can see on the board a circular ring like structure is shown here, and this particular arrangement of the cells in the form of ring is known as Kranz anatomy. What it is known as Kranz anatomy, where we will see that inside that is the cross section area of the leaves which shows vascular bundles, which is surrounded by cells called bundle sheath cells, and third layer which is called mesophyll cells. Now, so this ring-like structure, the arrangement of the cells in a ring-like structure is called Kranz anatomy, where we'll find that vascular bundle is surrounded by bundle sheath cells, and bundle sheath cell is surrounded by mesophyll cells, which appears as a wreath-like structure. You can see wreath, and it is in a form of a ring, and this full structure is called Kranz anatomy. So these plants, they exhibit this anatomy to undergo this process. Now what happened in C4 cycle, the plant which shows this cycle has few characteristic feature, as they have two cells going through this process. One is bundle sheath cells, another is mesophyll cell. You can see here they have few characteristic feature. First of all, bundle sheath cells, they have a granule chloroplast. What is the meaning of that? A granule chloroplast means they have chloroplast but there is no grana in it. Remember, so no grana present in bundle sheath cells, whereas in mesophyll cells they have a granule, like they have granule chloroplast, that means grana is present. That is why these kind of C4 plants, they have dimorphic chloroplast. So you can get in question of examination of NET and all, that dimorphic chloroplast means what? That is having both the category of grana, a granule and grana. So bundle sheath cells, they have a granule chloroplast. Along with that, they have thick walls. That means their wall that remains enclosed around the vascular bundle, they are little thick in nature, hence no gaseous exchange takes place in them. So no exchange of air, carbon dioxide or oxygen can take place in bundle sheath cells. But whereas in mesophyll cells, they have normal walls, so easily gaseous exchange can takes place. Third and very important point, bundle sheath cells, they have the enzyme called Rubisco. Now Rubisco you have come across in C3 cycle. Rubisco is the enzyme which helps in carbon dioxide fixation. That is, it binds with the carbon dioxide to produce the compound in C3 cycle, which you remember there that phosphoglyceric acid was produced. But here we have to see that bundle sheath cells, they have Rubisco, but mesophyll cells, they have another enzyme called pepcase. They don't have Rubisco in them. Hence, no Rubisco, hence no Calvin cycle will be performed in mesophyll cells. Will come later in the cycle. So these are some characteristic feature of the two cells which remain adjacent to each other, and they are connected by a structure called plasmodesmata. Now this is very important. It is a cytoplasmic connection between the two adjacent cells which allow the transportation of the compound from one cell to another. So what is the function of plasmodesmata? It allows the transportation of the compound formed in one cell to the another cell. Again back to the previous cell. In this way, plasmodesmata is a connecting passage for the two cells. Now what happened in this cycle? Remember, the cycle starts with 3 carbon compound in mesophyll cells. So in mesophyll cells, what is the compound PEP? It is phosphoenol pyruvate. It is a 3 carbon compound which combines with carbon dioxide with the help of enzyme PEPcase and forms 4 carbon compound called OAA, that is oxaloacetic acid. Now PEP, that is phosphoenol pyruvate, has 3 carbon, but combining with one more carbon dioxide, it has formed 4 carbon compound oxaloacetic acid. Now oxaloacetic acid is a kind of organic acid, and it changes or get into the next reaction, that is it forms malic acid, again another 4 carbon compound. It is another organic acid, and here breakdown of NADPH is needed. That means energy is required in this process, which is produced by the help of NADPH, which breaks and gives the energy. Now malic acid, which is formed here in the mesophyll cells, is transported to the bundle sheath cells through the structure called plasmodesmata. Now this malic acid which has come into the bundle sheath cells. Now we know that bundle sheath cell has rubisco, and rubisco has an efficiency to combine with the carbon dioxide to produce Calvin cycle. Now rubisco with the help of combining with the carbon dioxide, but before, malic acid is a 4 carbon compound. Remember, it has to release 1 carbon dioxide. So decarboxylation is taking place here in bundle sheath cells, where 1 carbon dioxide molecule comes out, and with the help of rubisco it enters the Calvin cycle, which forms the sugar or starch respectively. And 3 carbon compound comes out from here, that is pyruvic acid again is formed, and which travels back to the mesophyll cells for the regeneration of phosphophenol pyruvate. So this was the cycle where from pyruvic acid phosphoenol pyruvate is formed by the help of the energy which comes from ATP, which produces the energy by breaking down of the ATP. So what we have seen, that is a very simple cycle where we have seen that 4 carbon compound in the form of organic acid or oxaloacetic acid is formed in mesophyll cells. As there is no rubisco, no Calvin cycle could be performed in mesophyll cell, whereas bundle sheath cell has rubisco, hence it will undergo Calvin cycle. Remember, but there is no oxygen present in bundle sheath cell. As bundle sheath cell, they have thick walls, so no gaseous exchange is taking place, and hence no oxygen is produced there, but efficiency of carbon dioxide rises in bundle sheath cells by the decarboxylation of malic acid, hence Calvin cycle could be performed easily here. So one thing we have to remember, that in C4 cycle, as efficiency of cycle is more in respect to C3 cycle, as no photorespiration can take place in C4 cycle, whereas C3 cycle can undergo photorespiration if the concentration of oxygen increases in those kind of plant where rubisco is present. So rubisco combines both with oxygen as well as carbon dioxide. So if oxygen is more there, rubisco easily combines with the oxygen and photorespiration takes place over there in C3 cycle. That's a complete waste process. But in C4 cycle, no photorespiration takes place, hence it is a more efficient cycle than C3 cycle. So remember, C4 cycle takes place in tropical based plant like maize, sorghum, amaranth, where the climatic condition is really very hot, dry, and light intensity is very high. So remember, stomata most of the time remains closed. Why? Because stomata opening can release a lot of water, that is transpiration could easily take place, and also gases exchange could be easily taking place. So stomata most of the time remains closed in those kind of plants. This was C4 cycle, and very easy cycle to learn all the product. Learn it. This is very easy, and with the respective Kranz anatomy and the characteristics of both the cell, bundle sheath cells and mesophyll cells. Thank you for watching it. Please do like, share and subscribe my channel.